Hi everyone! Today's video will be an introduction to ultraviolet visible light spectroscopy. What is this? This is a lab technique utilizing a UV visible spectrophotometer to measure how much light a substance absorbs in a sample. This technique focuses on the absorption of light at different wavelengths. Molecules will have different absorption maxima. This is the wavelength at which they absorb the most. When we detect absorption, this detects the amount of energy needed to bring the electrons in the molecule of interest to a higher energy level. One molecule might absorb across a large spectrum of wavelengths. This is called the absorption spectrum. As shown here, the absorption versus wavelength graph for the specific compound is displayed. So how does UV visible light spectroscopy work? When looking at this schematic, we see that it is composed of a light source, a wave selector, the sample being tested, the detector, and a computer for processing. When choosing a light source, it is best to choose one that is able to emit across a wide range of wavelengths, such as a xenon lamp or a deuterium lamp. A wave selector, such as a monochromator, is used to separate light into a narrow band of wavelengths, and filters can be used to narrow down the wavelengths of light to make more precise measurements. A sample is then placed into the spectrophotometer to be analyzed. Note that it is important to also measure a blank or reference sample. To do this, we measure the absorbance of just the solvent inside which our molecule was dissolved. Once the light goes through the sample, it will go through a detector to convert into electrical signals that are readable. Detectors can be based on photoelectric coatings or semiconductors. A photoelectric coating ejects negatively charged electrons when exposed to light, causing electrical currents that is proportional to signal intensity. When passing through a semiconductor detector, an electrical current proportional to light intensity can pass through. This electrical current is then recognized by the computer. Now shown here is a more realistic drawing of a UV spectrophotometer. As you can see, the light goes through the wave selector into the monochromator and is then reflected off until it goes through the sample and into the detector to be analyzed. What can this technique be used for? This technique can be used for DNA and RNA analysis to determine its purity and concentration, pharmaceutical analysis to identify the compounds, bacterial culture to estimate cell concentration and track the growth at 600 nanometers, and lastly, beverage analysis to determine compounds of the drinks.